I've decided that not just to talk about my book that is just come out. Actually, it's only come out in the United Kingdom. It hasn't come out in the US yet. It'll be out early next year. But I thought I would give you a little background on what made me the kind of person that would write a book about animals and home ranges. And for starters, roughly, a home range is where an animal lives. So I'll change that definition as we go through the talk today, but that's where we'll start. Now, and now this is not working.
Does that to us. Plays games with our hearts.
Yes.
It really does. There you go.
There we go. Yes. So I'm on the faculty at North Carolina State University. I arrived there in 1979, stayed there through my active teaching career, and now for the past 15 years, I've been Professor Emeritus, which has allowed me to continue to work with graduate students, to teach students, and to do research through the university. My research started at a field station on Basswood Lake back in 1970. That's where I did my first research. It's where I took a course on vertebrate ecology and where I really got hooked on living here. That's where I met my first live weasel. Concy and I got married in the summer of 1972, and within a week, we were at the Rocky Mountain Biological Lab in Gothic, Colorado. I was doing research on coyote predation on marmots. But a female long-tailed weasel brought her brood of 13 youngsters up right in the middle of camp. And we had weasels everywhere. Concy decided she was going to catch one. She put out traps, and the next morning, she had one. It was a young male, and he ended up living with us for a year. He was a wonderful fellow. He taught us about the color change. He taught us about how weasels kill prey. He was just a wonderful guy. And over the years since then, we have had at least 7 weasels in our house at different times. 6 at one time, I believe. And we had 1 litter born in the house. At a later time, well, at a different time, I did some work on why weasels have black tips on their tails. That involved painting a big white patch on the top of the lion house at Brookfield Zoo outside of Chicago, and working with trained hawks that would attack anything that went across that white patch. And I showed that the black tipped tail actually distracts the hawks so that they miss the body of the weasel. I've live trapped weasels in a number of places. They're pretty fine fellows. I don't know if you're familiar with the wind and the willows, but in that, the weasels and stoats are the bad guys. Not long after being in Gothic, Colorado, Concy and I moved to the UP, where I studied fishers. UP is a pretty nice place to be. And I did some of my research on fishers up here out of the Kawishwe Field Lab down on the river. We hand raised two baby fishers that I trained to run on a treadmill so that I could measure metabolic rates at different running speeds. And from that, I could calculate how much prey they had to eat per day in order to survive. And I've continued to do research on fishers since. When I moved to NC State, I wanted to do research on weasels, looking at weasel home ranges, but I couldn't get money. The state, however, gave me money to do research on black bears. So I figured I could do research on home ranges of black bears. Now, handling black bears is not quite the same as handling weasels, especially big black bears. That work lasted well over 20 years. It was very productive. It was really fun. After handling a bear, we would move it to a place where it would be safe until it recovered from the drug. And then we left it. And this says late again, better be good this time. But now to my book that has recently come out. This is not a book that has a lot of natural history stories. It's a book that's been written for researchers to help give them ideas on different ways that they can approach understanding animals' home ranges. I'm going to run through a few of the concepts, the basic concepts that I cover in this book. But we'll start with an advance that doesn't work. Okay. Biologists have long thought that that animals do not wander aimlessly about in the woods, but instead confine their movements. And we can see references to that even in Darwin's Origin of Species. Elliott Coues in the 1870s noted that weasels settle into barns to prey on mice and that they'll stay in those places. He also noted that by killing mice, the weasels do more for the farmer than the sacrifice of a chicken now and then. And one of my colleagues recently actually showed that it's actually rats that kill chickens more than weasels, but the weasels get blamed. Ernest Seton Thompson, in his book, Wild Animals I Have Known, talked about the wolves that were in the drainage of the Currumpaw River in New Mexico and stayed in that drainage. Now, it's interesting that shortly after publishing that book, he changed his name to Ernest Thompson Seton. I have no idea why. But Ernest Thompson Seton in 1909 appears to be the first person who used the term home range in a publication. And Anthony, in his one of the first field guides to mammals, mentioned that most mammals stuck into one place or could be found in a particular place. William Burt in 1943 was the first to define a home range. as that area traversed by the individual in its normal activities of gathering food, mating, and caring for young. Occasional sallies outside the area, perhaps exploratory in nature, should not be considered part of the home range. Now, this is still the standard definition that most biologists use for a home range. And you'll see why I don't like it. First of all, he doesn't say why, really, or what an occasional sally is. Now, here Bruno is reading a letter from Sally that says, stop by occasionally. Now, this wouldn't be an occasional sally. This would be a very important part of Bruno's and Sally's lives. So I don't think it should be excluded from what an animal finds important. In the 40s and 50s, most researchers outlined live capture sites or observation sites and called that an animal's home range. Now, there are a couple problems here with this. One is that it ignores traps that have many captures. And one of my mentors pointed out in 1949 that what animals do inside their home ranges is more important than having the right outline. Other people decided that they didn't want to use straight lines to do the outlines. They wanted to draw curves, which does have a certain reality to it, but no two biologists could draw the same curves. By the 1960s, radiotelemetry was part of biology, and it gave us lots more data, and it allowed us to look to see where animals go when we don't trap them, and oftentimes when we can't see them. And now transmitters have become quite sophisticated, as have our ways of being able to follow those transmitters to see where animals go. But in the end, we still have dots on a map. We might have more dots. Each dot on this map estimates a location. The bright red is high elevation, and the green is a low elevation. And this, I admit, is one of my black bears from the Southern Appalachians. We can do a lot more with those dots now than biologists could in the 40s and 50s. We have ways of going from those dots to fill in the spaces between them so that we can see the areas that they ought to be familiar with. And I call this a untie my tongue. I call this a familiarity distribution, because it's a distribution of what the animals know. Now, one of the ways we do this now, fill in the areas, is use what's called a kernel estimator. And you can see over, you can see here, it's as though we put a little hill on top of each, we put out a map, put a little hill on each location, and then we stack those up on top of each other, molding them so that areas that are dark have the most locations by them, and areas that are very light have few locations. And that's how we build a familiarity distribution. The shape is actually a probability distribution of where the animals had been found. And one of the ways that we decide exactly how big to make this little hill has to do with what is the error associated with each location. Because telemetry tells us pretty much where an animal is, but not exactly. And so we can incorporate that error into this little hill. So here we are with Bear 61's locations from 1985. We had a pretty good system of being able to locate animals. We could drive our pickup truck up and down the Blue Ridge Parkway. That gave us a good overview of the area, and we could pick up telemetry systems from a long ways away, and we could locate bears 100 to 400 times in their active season. It's almost as good as GPS telemetry. And here we have an outline, a contour outline of Bear 61's familiarity distribution. Now, notice this notch in the familiarity distribution. The first time I mapped out, I actually plotted the dots. I saw this notch and I said, I know what 61 was doing. So you can see where Mount Pisgah is. That's the high spot, about 6,000 feet. Little Pisgah's just a little bit less than that. And going down from Little Pisgah is Little Pisgah Ridge. Now, that's not just a regular old ridge. Little Pisgah Ridge is murder on anybody going up and down. It drops 3,000 feet down to Big Creek drainage. And going up, you can't do it without sweating like a pig. And going down, you can't do it without your thighs telling you to stop. There you see it. And 61 didn't want to go over Little Pisgah Ridge. She preferred to walk around. That makes a lot of sense to me. But here, the map shows us where Bear 61 was familiar, but it doesn't tell us why she avoided Little Pisgah Ridge. I knew that because I'd been there so many times. But this is one of the problems of just a descriptive familiarity distribution. It shows us what, but doesn't really give us insight into why unless we've got other information. It doesn't show us what 61 knew about her world. So let's look at this from a couple other stories. This is a map that shows my family cabin in northern Wisconsin. And I became very familiar with that land over the 65 years that we owned it. But this map doesn't communicate to me what was important about that place. So I'd take off from the cabin, I'd hike down the ridge, and I'd walk by places where I knew I cut oak for firewood, and I knew where we had a jack pine, jack pine, red pine plantation, and where I had to thin the trees. And then I'd walk on down Heart Lake Road, and I'd end up by this wonderful bog. It smelled wonderful. Lots of black spruce, lots of Labrador tea. But the fishers that lived there, they didn't see it that way. They were interested in other things. A fisher coming by would know where the oaks were, but wouldn't really care. But she'd care about the voles, the redback voles that were in the forest. And then she could smell any carcass that was within 2, 3, 400 meters, 2, 3, 400 yards of where she was walking. And she also knew where there was a rest cavity in a big oak. And then she'd head down to the bog, and that's where she hunted snowshoe hares. That's the way she saw our place. And a bear would probably walk up the driveway to get to the trail down the ridge, and it could smell carcasses just as far away or further than the fisher could. And it was interested in the clear cut more than the bog. And it knew where the blueberry patches were, how fast they renewed, and it also knew that coyotes and foxes and raccoons were also interested in those berry patches, and that it had to keep track of those. So one last example of this kind of thing is that in 1990 and 91, Constantine and I were on sabbatical at the University of Wyoming in Laramie, and over 66, 67 days, I marked down everywhere I was, everywhere I went, to the nearest 3 meters. That's about 10 feet, oftentimes much, much more precise than that. And I knew exactly where I was every minute of those days. Now, this is the map I drew. And every line is someplace that I went, and the darker the line is, the more times I went there, and the more time I spent there. There's a dot up here for my house, very small but very black, and a dot down here for the biology building at the university. And then you can see all of my travel routes. Now, I took these I took these data, and then I took a sample the way one would from radio telemetry or from live trapping or anything like that. And I made a familiarity distribution to show what a biologist would do who took a sample of my movements. And you can see this familiarity distribution looks a lot different than where I actually went and what I actually perceived. I did not consider people's houses to be part of my familiarity distribution. I walked mostly, or rode my bike, or drove our truck on roads or, confined areas. So I'm not really happy with being stuck with familiarity distributions like this to understand animals' home ranges. Different animals view the world differently. Here is how I see a view. Here is how I see a view upslope from our house in the wintertime. And that's how wolves see it. That's a lot like what we would see, but it's more from my waist. This is what a weasel sees. Same place, trying to deal with them in exactly the same way biologically just leaves me worried. The local red squirrel knows these pines differently than does the local black bear, both of whom go through that area. And the red squirrel even knows those pines differently than the local flying squirrel. So where animals go determines what becomes familiar to them and to what their home ranges are. But what is familiar to them then dictates where they're going to go in the future. So you get a feedback that the home range actually comes from the movements, but then the movements are dependent on the home range, which is what the animal knows. So this is one way to look at it. This is what a familiarity distribution tells you. This is what's in the critter's head. And this leads us to the concept of a cognitive map. This is actually something that's housed in our brains and in all the brains of vertebrates and a lot of insects. We know that it's in mammals and birds and all the vertebrates, it's housed in the hippocampus, which is tucked in the brain. And it's kind of a cognitive control room. It helps critters. It's basically, well, let's go to the bottom of the slide. It works like a card catalog in an old library where you could, work through the card catalog, and it tells the brain where to find a memory, and then it helps the brain plan how to use that memory in the future. And it's not just memories, but it has a lot to do with planning. It's important for navigation. It's important for social interactions. So in a way, if you think about it, a social interaction is a way of navigating through dealing with people. The hippocampus deals with the problems of interactions and how to take ideas and move them back and forth. And my definition of a home range is that part of the animal's cognitive map that the animal keeps up to date. And it includes places that aren't visited recently, but that are held in memory for future specific uses. That latter is important. Working on my black bear project, there was one day when one of our females picked up and disappeared. I think it was female 260. She just disappeared. We couldn't find her. And finally, one of my graduate students drove way down the Blue Ridge Parkway, way beyond where we normally would have to drive. And he found her signal. And she was way down there, and she'd taken her cubs along with her. She stayed down there for a few days. Then she turned around and came back. I'll wager, that was a cruddy year for acorns. And I'll wager she remembered that ridge way down the parkway. Maybe her mother took her there, but she remembered that. And Sue Mansfield, working here with Lynn Rogers bears, documented the same thing. I think, I can't remember whether it was Dot or Donna, but headed way up the Echo Trail. And Sue went up there and found that where she'd been hanging out, where the bear had been hanging out, there was a nice ridge up there with oaks. I mean, we don't have very many oaks around here, but she remembered that and she took her cubs there. So that's one of those occasional sallies too. And it's important. So how do we really know that animals go through these cognitive gymnastics? to do all this with their hippocampus? Well, one way is with brain scans and working with critters in the lab. These two two illustrations here show what are actually are digital slices of parts of the hippocampus. And you can see that on both of them, the dark colors represent where cells in the hippocampus have lit up. And they light up actually as a map of where the rat walks around in its cage. And on the right-hand side, it was a round cage. And in the lower right, that's where the rat spent most of its time, because that's where the cells lit up the most. And in the one on the left, it was a rat in an X-shaped cage, and it stayed in that arm in the lower left. The hippocampus has these are called place cells in the hippocampus. Hippocampus also has grid cells. It has time cells. It has head direction cells. A recent paper just came out in the past 2 weeks showing that Egyptian fruit bats that were carrying it had electrodes entered in their heads, and then they carried a small package of memory that the biologists could pick up later. But those bats were flying around on an island, and the head direction cells always showed which way was, well, I can't say north, but what was most important for those bats. They had a direction that they were able to keep, and it was the head direction cells in the hippocampus that showed where they were going. And those same kind of fruit bats plan trips to fly out and fly back to different food sources. And while they're flying, they think ahead, and that can be picked up by the electrodes. They think ahead of where they're going to go, but they also think back of where they've just been, and they can put that in memory. And so this back and forth in the hippocampus is going on all the time. I think this is really cool. Now, another way of thinking about these cognitive gymnastics is to ask the critters directly. Has anybody in here read about Stella? Oh, Polly has. Stella is the dog that's owned by Christine Hunger and her husband. And Christine is she works with kids who have language problems. And she noted that when she and Jack got their puppy, Stella went through in her communications with Christina and Jack, Stella went through the same stages as she developed that kids with language problems go through, and actually all of us go through. But with the kids with language problems, Christina had this tablet, and kids could press buttons to show images that would then show what they wanted to discuss or what they wanted to communicate, and the tablet would say the name, and so the kids could communicate that way. She thought, I wonder if I could teach Stella to push the button. And so she got these buttons, and sure enough, with a lot of patience, she could get Stella to push. The first button was outside. But now, at the time the book was published a couple of years ago, Stella had over 40 buttons that she could use. And they weren't all nouns. You know, there were verbs, there were adverbs. So she could even make rudimentary sentence structure. She could communicate. And she could say, after she got back from a walk to the beach, for example, could express information about that trip to the beach. And it showed that while on that hike, she was thinking ahead of where she was going. She was thinking back about where she'd been, just the way we see it in the hippocampus. That's cool. And incidentally, a number of other people have jumped onto this bandwagon and are teaching their dogs to do this. And the third way that we can look at these cognitive gymnastics is to do science. Now, I think of the scientific method as this spiral that goes up. And one starts in the lower right with observations, things that we think are pretty cool. But we want to know why animals do this. Well, that leads us to generate hypotheses. An hypothesis is a simple statement of one possibility for why an animal does something. And the goal then is to prove that statement is false, or to show that, at least for now, it appears to be true. So once we have hypotheses, we have to design research that lets us test those. We collect data, we do analyses, that leads us to results. And then the results become an observation for the next loop around the scientific method. Scientific method was one of the neatest ways of showing us how we can learn new things and to have confidence in what we've learned. Now, thinking about a cognitive map, and we want to learn about it, we can guess, we want to know how a squirrel will move through this tree. The cognitive map keeps track of where the squirrel is, where it's planning to go, and it tells her how to get there. The cognitive map in the hippocampus has jumping within its memory, for example, and it tells the squirrel how hard it's going to have to jump in a given place, and how it's going to have to land, and how much the branch will bend where it lands, and what the bark will feel like in that branch, because all that is in its memory. And the squirrel uses this to plan its foraging routes or escape routes and how to time them. So if we go back to the black bear that wants to go to the berry patch, after it's left the berry patch, it knows that those berries, the berry plants, are still there, and they are going to start renewing. And so it has to figure out, when should I go back to that patch? And this is where I start getting into making models to help me understand what I think animals might do. So here, the bear has information about that blueberry patch. And if you look down here, this is how the food renews as time passes. So the x-axis is how long it's been since the bear was last at that berry patch. And the y-axis is what is the berry abundance in that patch. But at the same time, the longer the bear has been away from that berry patch, the information it has about that patch decreases. There are going to be other critters that come and might take berries. It might be that the berries don't renew quite as fast as the bear had thought they would. It might be that what it had learned the last time it was in that patch about where the best bunches were is something that's changed. And now later in the berry season, 10 meters away is a better place, but that would slow down how fast the bear can forage. So you can see we have two things going on. One is the berry abundance coming up and information about that abundance going down. And we can plot those on this graph, and that will show the best time to return to that patch. And that's when the bear should come back. It should not wait until it should not wait till the abundance has maximized out. It should come back sooner so that it can do a better job of foraging there. Now, we have good berry years and we have bad berry years. And we can plot the predictions on a graph. And you can see that in a good berry year, the bear should come back sooner than in a poor berry year. So we have an hypothesis that has been generated by the observation of my model. And that says that in good berry years, bears should return to berry patches sooner than in poor berry years. Well, what do real bears do? Here's what they do. They actually do return sooner in good berry years. Here you can see this is the time and days to return, and the higher the hard mask, the nut index is, the sooner the bear returns. Now, these results lead to two new hypotheses. One is that autumn food is way more important than summer food for the bears, and that we need new hypotheses. We need to develop new hypotheses that will explain why there is so much variation in these data. It's clear that the bears are not responding only to the berry crop. They're responding to other things as well. And we have the same sort of result for fishers. And that leads us to two new hypotheses. One is that gray squirrels are way more important for fishers than are red squirrels and ground squirrels. The gray squirrels are the most important for the fishers, and these data are from my research in Northern California. And again, there's a lot of variation around that general trend. And we need to figure out new hypotheses that might help explain why the return times vary so much. And here's another example of how we might use models. Red squirrels defend individual territories. And in each territory, there is one central food cache or a few central food caches. Turns out that in the Rocky Mountains and the Western Cascade Mountains and California mountains, red squirrels tend to have a single cache where they cache everything. But around here, for some reason, the red squirrels have multiple caches. They're all pretty much in the middle of the territory, but they have several of them. Nonetheless, when a squirrel has got this cache, it wants to get food into that cache to help him or her make it through the winter. And that means that for here, they have to go out to all edges and everything in the middle in order to get their food, and they have to bring it all back to the middle. Now, why don't a male and a female squirrel share a territory? Well, if they did that, they'd have to have twice as much food in their cache. That means they'd have to be able to forage over a large area. They'd have to increase the area where they forage. And that means they'd have to make trips that are further out, and that means they're spending more energy. So this is what the model says that red squirrels should do. It says that it's really more energy efficient to have individual territories instead of shared territories. And indeed, that's exactly what red squirrels do. But we can generalize from this, and it is that animals maintain individual home ranges unless two or more individuals are needed to raise the kids, to get food, to repel neighbors, to build cavities, to pine boles in the southeast that I studied maintain tunnel systems. And it takes a couple of the voles to keep that tunnel system in good shape. And if we look at it, this is not new information, but the hypothesis is consistent with what black bears do, like red squirrels. Each has its own individual territory or home range. And wolves, which need help to capture prey or to repel neighbors, they share territories and live in groups. Now, I'd like to end up with one more story, actually, but it shows it something a little bit. It shows a different type of modeling. But when I was doing research on fishers at the Quishui Field Lab, and it was wintertime, we'd be inside in the winter. There are a couple Forest Service guys there and graduate students. So I was sitting in the main living room, and I noticed there was a weasel that was running along the wall, and then it came out from underneath an overstuffed chair on the other side of the room from me. And I got up, went into the kitchen, sliced a little piece of venison off a haunch we had there, came back, sat in the middle of the floor, held that venison out towards the weasel, and the weasel slowly came up. Oh, its tail was all fluffed up. And it'd take jerky steps, you know. And then it finally got to me, and I had to, you know, I just sort of held myself there, told myself that if he bites, I'm going to have to just hold still. And the weasel came up and it head butted me. It hit my fingers really hard with its muzzle. Completely surprised me. I dropped the venison. Weasel grabbed it, took off underneath the chair, and ate it. Well, I felt pretty stupid. I went back, cut off another piece, came back out. We went through the whole thing again. He headbutted me again, and I dropped it. Now I really felt stupid. So I went back and got a third piece, and this time I held it. He did headbutt me, but I didn't drop it. And he settled in just to eat it right there from my hand, which was pretty cool. And ultimately, I let him have the last little bit. Now, we can actually model that kind of an interaction with the type of models called game theoretic models. And we can look at it as though the weasel and I were playing a game. And the winner was the one who either got the venison or got to hold it while the weasel was there. And that kind of modeling can lead to lots of different hypotheses. And there are other kinds of modeling as well. And I try to cover a bunch of those in my book to give people ideas on different ways to understand why animals do what they do. And that's the end. And I've gone a little bit over. I'm willing to take questions. Yeah.
I'm happy to help.
Connie drew a lot of those pictures, didn't
He sure did.
Yeah.
Yeah, she illustrated the book, and as the cover says, she was some, yep.
Okay. Lovely.
Congratulations. Yeah,
Beautiful.
good work. Yeah, Bill?
When you said gray squirrels were more of a prey of fishers than red squirrels or
In California.
in California. So were you talking about, is there prey adults? Or does that include young?
Gray
in
squirrels?
cavities or?
Fishers can't go into cavities. They're too big for gray squirrel cavities. If gray squirrels are in leafy nests up in the trees, they can go in there, but the squirrels don't usually have their youngsters there because predators can get to them too easily. And he
More accessible?
they're bigger is the biggest thing, I think. Yeah.
When the bears are checking out the blueberry patches, do they have any idea of how many other berry pickers are out there?
Oh yeah. Yeah, they can smell if nothing else. And they can really do a good job of smelling who's been here and for how long.
How many?
And
So they can get a good idea of how productive raccoons or whatever it is?
Yes,
Oh.
yes. And there's another part of the model that I didn't show, which is that just as there is variation from year to year in berry production, there's differentiation from year to year or even shorter times than that in how fast the information is lost. So if a bear comes into a patch and it knows that, man, the foxes and raccoons have been here, it knows that it's going to be losing information faster than in another year when it can tell that it doesn't have that kind of competition.
Good.
Yeah.
Very interesting, all of this. I just wanted your impression of something. We live on Sunset Road, and Thursday night we were driving home in the dark and we saw a bear across the road. The bear went probably, I think you estimated 30 feet off the road and just stood and scratched on a tree. We stopped our car, okay? And it was dark, so our lights are on. We're right there watching it. It went up the bank just a little bit, scratched on a tree, and then hid behind like a few twigs. I mean, we could see the bear in the dark. And we sat there for quite a while and just looked at it, kind of talked to it, just kind of thought it was the coolest thing ever. And it didn't go anywhere. It didn't climb the tree. It didn't run into the woods. It just stood there. And Gary said maybe it was a mom with babies and it was staying close, but I just wonder what's your impression of that? We thought it was the coolest thing ever.
Bears scratch trees and they also bite them, usually as high up as they can reach. And they'll stand on their hind legs and rub their shoulders and their backs on the tree. And this is all scent marking to tell other bears, hey, I've been here. And the other bears then know from the way that the scent dissipates over days, how long it's been since Jane was there, or Jack, whatever. And the bear may well have been just waiting. It might have wanted to do more scent marking and was wishing that you would leave so so it could get out and do what it wanted to do. The other thing is that bears are slowing down now. They're going into their dens here about now. And where I studied bears in the Southern Appalachians, we figured they were almost always all in their dens around Thanksgiving. But here they go in earlier.
Thank you, Roger.
Thank you.